Hi everyone, here it is, week 7. I'm Elizabeth Morin. It is February 25th, 2018. This week we are doing the dissection of the sheep brain. To start off, we're going to be removing the meninges. It's going to be this thick layer of protective tissue that, and I kind of got a start on it for time purposes, so, because it is a very thick tissue layer. So once this gets removed, we can begin pointing out the different structures of the brain. And there we go. We have our brain. And we'll remove the meninges right there, set it aside. And we're going to be starting off with the ventral side of the brain, the underneath side. So here we go. We're going to go with our cerebellum is right here. The parietal lobe. The mammal body is going to be right in here. The optic chiasma is going to be right here where these were. And actually on the meninges you can see that they were removed, the connectors that were right there. From there we have our olfactory bulb. It's going to be right here. That's right here. The optic nerve is going to be right in here also, and that also was right in here. That kind of got removed right there. It's going to be this piece right here. That's our optic nerve. Optic tract, I would say, is going to be right here. The midbrain, right here. The meninges, most of it got removed, but this was also the meninges right here. If they were not removed all the way, it would have been right here, and you can see this area where the gray matter is, and right in there. We have our pons. Let's see, I'm trying to see. Yeah. Right here would be our pons and our medulla oblongata. And that is for the underside of our brain. And we're going to flip it over and we're going to go to the dorsal structure of the brain. And here we have our cerebellum, right here, our occipital lobe, our parietal lobe, right here, longitudinal fissure is going to be right here, this part right here, our frontal lobe, these are our frontal lobes right here. Our sulcus, gonna be right in here. The ridge right here is gonna be the gyrus, and our temporal lobe right here. Now we're going to need to go into, we're going to separate these two parts. We're going to pull the cerebrum away from the cerebellum, and we're going to look for some nerves in here. We're going to see in the glands. Let's see, look right here, the lighting's kind of bad. Right here it looks like we're looking at our pineal gland, and our right here would be our corpora quadrigeum right here. And then course, we have our cerebellum. All right. Also want to go and show you the nerves. They're in here. You can see some right in here. And some right here. There's one right there. And also, you can get in here, there's some nerve right there. Okay, now we are going to cut along the midline and produce the mid-sagittal section. So I'm going to cut right down the middle. It's a lot easier to cut than I thought it was going to be. And cut down the cerebellum right here. And down the spinal cord. Okay. And here we go. Now we have two halves of our brain. I'm going to pause for just a second and get a still view picture of this. Okay. All right. So now we're going to start off and go over the different sections of this. I'm going to move one aside so I get this other one out of the way. Okay. Okay. Well, we're going to start off with the central canal right here. And then, of course, this is our spinal cord. And the only difference is because this is a sheep that it's going to be going straight out, where if this was a human brain, it would be going vertical with our body instead of horizontal. So, and then we're going to see right here is going to be the medulla oblongata. And then our pons should be right here, midbrain, the mammillary body. Then we have our hypothalamus. And then right here, I'm looking at this, let's see. Let me study this for a second. All right. Here would be our corpus callosum. And then our optic chiasma. And then our thalamus. And then our, right here, our pineal gland. Our cerebellum. And our white and gray matter is actually in here. Our arbor vitae is the white matter, and then you can see in between it is the gray matter. And if you can pull it apart. I think it's a little discolored now that it's been sitting for a little bit, but there you have it. That was that was the dissection of our sheep brain. And thank you for viewing, and we'll see you next week.